spleen samples, right? So here, what we're going to do is we're going to look at relative expression of the TTR gene in liver, heart, and spleen tissues. We want to compare the three tissues, and just like you would do if you were thinking about something like an RNA-seq experiment, where you want to look at, you know, the expression level in this tissue versus the expression level in this tissue, we're doing the same thing here, right? We can compare and say expression in this tissue was X times more than expression in this tissue, okay? The way we do that is through a calculation called delta-delta CT. Okay, so here's the math portion of the summer workshop. I know there's some other math portions, but this is a math portion. But it's an easy math portion. No big problem. The first thing we do is we calculate a delta CT. When we talk about delta CT here, right, we're talking about comparing our gene of interest, the TTR gene, to a housekeeping gene. So you always need a reference gene if you're going to do this. So you look at expression level of something that should be expressed uniformly across sample types. So we want some sort of housekeeping gene, right? Something that's expressed at background levels across tissue or sample types as our calibrator or our equalizer, essentially, for our samples, okay? So in the case of this experiment, we do the 18S gene. We run this ahead of time. Again, this is not what you would do if you were doing this experiment for real, okay? If you were doing this experiment for real, you should be assaying for your housekeeping gene at the same time on the same plate as your experimental gene. Okay, you want to minimize variation by running both at the exact same time. So that way, if the instrument is behaving different that day, it's captured in both of those results, right? You're still comparing apples to apples, okay? We don't do that here for the sake of making this a little bit more manageable for us to get through and just to handle in the process. But that's what you would want to do. But for the purposes of understanding how this works, it's exactly the same. Okay, so the first thing we're going to do is we're going to look at our CT values, so our cross threshold. CT means cross threshold or cycle threshold. When we looked at this previously before dinner, right, we talked a little bit about, and I think Steve did also somewhere, I'll put it here. Yeah, right, we set a threshold in the early exponential, right? So what we're doing is we're looking at the cycle on the x-axis at which our sample, its fluorescence crosses that threshold value. That's our CT value, okay? And so we're looking at that for both our gene of interest, okay, which is what you see in the plots in front of you. So the plot's not in front of you. The plots that Kalani emailed you. Those have your liver, heart, and spleen amplification curves on them. And we're comparing that, we're looking at the difference between that and our CT value for our housekeeping. Okay? So here is the average CT value for liver, heart, and spleen in our CYBER experiment and in our TACMAN experiment. Okay, so these are the averages of all of your samples in these experiments. Okay, we just averaged them all together. So think of these as biological replicates, right? You all ran a biological replicate. We're taking the average of those biological replicates. What I have here is I tried to color code for you. I don't know how helpful it is, but what we're going to call the normalized CT, or the difference between our sample CT, our experimental, our TTR CT, and our 18S housekeeping gene calibrator CT. So for liver, right, we had 8.8 as our CT average for the calibrator and 13.6 as our CT average for the TTR in liver. So your samples crossed the threshold on average at 13.6 in the CYBER Green lab, the mutant lab, for the liver samples. And that results in a difference of 4.8. Okay? So that's our delta CT for our normalized CT that we've normalized against that that housekeeping need. Okay, we can do the same thing for all of our samples. So average CT and our calibrator CT for our difference, average CT and our calibrator CT for our difference, and then the same thing for the TACMAN index. Okay, so no difference in the analysis between Cybergreen and TACMAN, exact same process. Good so far? Not too bad? Okay. The next thing we're going to do is we're going to pick one of these as our, so I always confuse the language on this. The proper way to say what we're going to pick next is the calibrator. I talk about this as calibrating because we're normalizing, right? But what we normally call the next one, or what we pick next, is our calibrator. So I'm sorry if I say the same words in the wrong places. But basically, you select one of your 3 samples as your calibrator. So you're gonna say, this is the one we want to compare against, right? You have a control sample and then you have a few experimental conditions or whatever you're doing. Maybe we want to see, you know, we're interested in how this, we know what the expression should be in spleen, and we want to see how liver and heart compare, right? So spleen becomes our calibrator, and liver and heart are what we're looking to find out, okay? Doesn't matter, you can pick any one of them, right? It doesn't change the results, it's just what you're comparing to, okay? So we're gonna pick spleen as our calibrator. And so the next step here is we're gonna set a calibrator, right? We're gonna use spleen, and then we're gonna calculate our delta-delta CT. Okay? To do this, we take the delta CT of the sample of interest, the liver, heart, or spleen, so what we just calculated, and we subtract from that the delta CT of our calibrator. Okay? So in its simplest form, right, we're taking what we just calculated for spleen and subtracting it from what we just calculated from spleen. Okay? So spleen minus spleen equals 0. Right? If we look at heart, we have a lower CT here, so 13 minus 16.5 is negative 3.5. And same thing here for liver, right? Negative 11.7. And then we have our results down here for that.
What is the object of this? Because we have calibrated in the first place, and now we are calibrating against one of the samples.
Right, so in the first place, we calibrated against the housekeeping gene. Here, we're going to be able to determine how many more, the object is to figure out what fold expression change we have in 2 of our samples versus a 3rd. So we can hold, essentially think, right, so we're holding this as our comparator, like what we want to compare against. And we're going to determine what fold change difference we have for TTR expression in these 2 tissues. So there was 5 times as much expression. There was 100 times as much expression.
But is there a justification to take one of them, or?
Just what you could do any of them, right? You could set this is your calibrator, and then we do spleen here, because spleen is the lowest. So we're gonna say, this is the lowest, and this one is this much more, and this one is this much more.
Oh, it's spleen is the highest.
Spleen has the highest CT value, but it's the lowest expression. Remember, CT values are, there we go. They're inversely correlated. If you cross threshold here, it means you built enough fluorescence to cross the threshold earlier, which means you started with more, right? So later CT means less sample, less original sample, because it took more cycles of exponential amplification to reach the detection threshold.
So it's a simple interpretation of this that the gene is expressed in the liver 3,300 times the level of spleen.
That's a simple interpretation based on the cyber results from your experiments. Yep. Right, so for every copy of TTR that's produced in the spleen, there's 3,326 copies of the TTR gene product produced in the liver. Right, we can't say protein because this is at the level of messenger RNA. So, right, there could be post-transcriptional, effects that happen. We don't know any of that. But they are making, the liver is making 3,326 times as much TTR messenger RNA as the spleen is.
Why is a bit different? You see the pattern?
Yes, the reason it's a bit different is because when we run this many samples with a class of people, there's a couple of mistakes, and the mistakes mean that the results are, right, are going to be different. So, you know, you can, I'm relatively certain there was one group where, we won't point out groups or anything like that, but where samples got switched, right? We called one thing spleen and it was actually liver or heart, I don't remember, but, you know, what we thought was one thing was another, because we had fantastically early amplification
Yeah,
for
no, I
spleen
got it.
and much later amplification for something else, right? So, and I didn't correct for that in here, because I wanted to be able to talk about it. And really, what you can do after we finish this, if you want, is you can do for your individual, right? You can calculate the fold change for your sample independent of everyone else, and see what you get, okay? You know, this isn't, I wouldn't even go so far as to say we should trust any of this data, because, like I mentioned, we ran your housekeeping genes separately. That is the wrong way to do this, right? If you submitted this for publication, first of all, they would say, why are you telling us this? This isn't important. But then secondly, they would say, you didn't run your, you know, your housekeeping gene on the same place. This is garbage data, and it's really good, right? Which it would, okay? But for the purposes of understanding, it works just fine. Okay, last step in this, to calculate, so to go from these numbers here to our fold change, right? We do step 3 here, which is to determine the relative expression based on that number that we just saw, okay? And to do that, we raise 2 to the negative delta delta CT. Nope, 2 was where we compare against the calibrator of the spleen. So step 1 was against the housekeeping. Then we take the results of that and subtract the calibrator, which we set to spleen. And then 3 is to take that result, right, this number here, and plug it into this equation. So our delta delta CT is that number. We raise 2 to the negative power of that number. And what that tells us is our fold change, right? So for spleen, it's 1, because it's spleen. Yep.
I know there were some mistakes and it's like noisy, but I mean, there's clear differences between the CyberGreen and the Pac-Man. One is saying it's 3,000-fold difference, and one is saying it's 16-fold difference. So there's like an enormous difference between what these two techniques are giving us.
But it's exponential, right? So think of how few cycles you actually have to be off to go 16 to 3,326, right? It doesn't take, with a relatively small number of samples and single replicates and 1 or 2 very different results, we get off pretty quickly.
It's very hard to say things concretely, quantitatively about these differences.
Absolutely, based on this, right? If this was your results and you looked at your data and you saw that, you know, again, right? But, right, for the purposes of seeing this and understanding, I think it's actually constructive to see the change in, or the differences here, because it doesn't take a lot for it to become very problematic, right?
The assay technique is Pac-Man more with like reliable.
Tachynan is more reliable in terms of its chemistry, in terms of minimizing off-target amplification, but I don't want to say more reliable, because if you have a well-optimized CyberGreen assay, it works beautifully. You just have to understand that you have to watch out for primer, dimer, and off-target amplification, and run your melt curves and make sure you get that nice peak where you think you should have it. If all that falls into place, there's no problem with Cyber, right? Cyber works fine, it's just, there's more to consider and more to work out to have a Cyber assay that you can be confident in the results of.
I think the values are flipped from the sheet. I think the Cyber values on the sheet are what we ran off the Pac-Man.
Are they?
Yeah.
Okay.
And the Pac-Man ones are not the same as the ones here. Yeah, the numbers
Uh-oh,
are
is this
pretty-
last year? I wonder if I didn't change all of it. Okay, well, I can try and rework this and get it to you, but everyone understands the purpose, right?
Yeah.
Right, so it's not honestly so much about, unless any of you are researching TTR and liver heart. Probably. All right, and so that's all I have for this. Does anyone have any questions? Right, so it's pretty straightforward, the way we generally analyze this. This is something, when when RNA sequencing was first a thing, right, you would always have to do this to validate your RNA-seq fold change results. That's become less and less common, right? Generally now, RNA-seq data is trusted at face value without having to do a lot of real-time validation of your RNA-seq results. But if you're interested in one or two genes, and you know what genes you're interested in, right? If you don't know what's upregulated and what's downregulated between tissues, and you're interested in sort of blanket changes in expression, you're doing next-gen sequencing, right, RNA-seq. If you're interested in TTR, you're doing qPCR, right? Because it would be a big waste of money to do next-gen just to get information about TTR.
Can you go back to the slide with step 2 and the second notes that
All right, anything else? We are early. I don't know if we can be early. Do it again.
That's right, Ray.
We're like, so I have to do protocols, but protocols is gonna take about 35 seconds.
Yeah.
Because you're really doing one thing downstairs. What I'll do is we'll do protocols, and then I'll give you like a 3-minute break while I just run and check to see if they're ready for us.
Okay.
And if they're ready for us, we can go down. If not, we'll either have to hang out, or if you want, I can talk about something else like digital PCR. But I don't know if I really want to start digital PCR.
Can you go to step 3?
What's that?
Can you go to step 3,
Step 3, yes.
please? Thank you.
Okay, so, yep.
One quick question. So